Hello, my name is Katrina Exter. I work for the Flanders Marine Institute in Belgium, and I'm going to talk about data management for the European Marine Omics Biodiversity Observation Network. EMO is a long-term marine genomic observatory. We perform regular standardized sampling of the marine environment, standardized sample processing, and produce standardized sequences and bioinformatics results from the samples we collect. We have 17 partners covering the European coastline. The network was initiated and is run by the European Marine Biological Resource Centre to support policy needs and the sustainable use of marine resources. Now, EMO is a data-heavy enterprise. We collect sample and event metadata, measurements, sequencing data and metadata, and bioinformatics data and metadata, as well as a lot of associated data. It is important to us that all these data are fair, open, and of high quality. We also require that the workflow that does the data management is inexpensive, can be run by anyone, and produces outputs as quickly as possible. So clearly we needed to build our own data management workflow, repurposing as many existing resources, technologies, and specifications as possible. We began building this workflow in 2021 when the project started. There were a number of challenges we faced when designing the data management workflow, and these all can be summed up in one word: diversity. Our inputs are diverse. EMO data consist of sampling event log sheets, sequences produced from the collected material samples, bioinformatics results produced from those analyzed sequences. All these data come in on their own timescales, but they all have to be linked to each other so they can be explored as a unit. The providers of these inputs are also diverse in location and in their background, in their level of data literacy, and therefore in what kind of data handling approaches they want to use when interacting with the data management workflow. And the formats, standards, and types of data we are getting are diverse. Our outputs are also diverse. We have to focus on the human users and the machine users of our data. We need to allow our data to be accessible to many different types of community repositories, as well as via our own database. And we have users with different levels of scientific and technical needs. We have several chains of command working on the different aspects of the data management and who need to be part of the workflow. We have the bureaucratic people, the people dealing with EMO policy, the technical people, and those making the scientific decisions. And finally, we have to deal with entropy, diversity increasing with time. Now, EMO itself aims to produce samples and data that are comparable over the entire lifetime of the project, so changes to the procedures and protocols will only be done if absolutely necessary. But the data management should be able to accommodate changes in the way data are produced and consumed. As technologies and expectations change, the data management must be flexible enough to accommodate this. So what solutions did we come up with to solve these diversity challenges? I'm not going to go over the entire data management workflow, but I will highlight a few features that are particularly important. First of all, we decided to centralize the data management in our EMO GitHub space. We needed a central location so we could do the quality control, semantics, linking, and reformatting in one place. There are many reasons we chose GitHub. It's free, shareable, widely used, flexible, version control, can run actions, etc. But I will add that we're not beholden to GitHub itself being available forever. We rely more on Git than GitHub, and we can move to another centralized location if we need to. Secondly, it is very important that we have a low entry threshold for the data providers of EMO. This goes especially for the scientists at the sampling stations. These are busy people who collect samples and data for many projects, and they don't have the time or the expertise to do different complicated data collecting actions each time. So we use Google Sheets as our log sheets, as these are familiar to everyone, and we format them to minimize incorrect data entries as much as possible. After pulling the log sheets into GitHub, we carry out an automated QC workflow to check for errors, and by including a feedback cycle with the station, the number of errors slowly drops with time. Next, we add the semantics to the data. For example, in the log sheets, each value in each cell is linked to a controlled vocabulary for the type of data, the type of value, the measurement unit, etc. This is done automatically using GitHub Actions and combined with our data model, in which we have described all the parts of the EMO-born data world. This allows us to turn these spreadsheets into a machine-readable and machine-understandable format, being RDF, Resource Description Framework, which is a standardized way to represent linked open data, LOD. Now, I'm not going to explain RDF and LOD, there is no time, but note that these are the framework and the language of the web, and by using these, we can make our EMO-born data not only findable via the web, but they become part of the web. Next, we group our data into packages called Research Object Crates. RO crates are an approach to packaging data files that belong together. The files can be located in one place or many places, one GitHub repository, or spread over different community of repositories. An RO crate package includes a metadata file that describes the contents of the crate with rich and standardized metadata, and so that the relationships, the links between the files are also described. This allows one to group together with metadata and in a machine-readable and machine-understandable way, files of disparate type and files of disparate storage location within a single package. So the log sheets, the QC outputs, and the transformed into RDF log sheets from each individual observatory are each one crate. The sequence metadata for all sequences from all samples are another crate. The outputs of each bioinformatics run on a sequence are also a crate, and so on. Next, we use GitHub Actions to have automated workflows that process data and metadata in whatever way is necessary. The more automation, the fewer errors, and the more speed. And finally, we want to make the EMO-born data accessible not only via the GitHub site, but also via a more familiar web interface. Our data RO crates are viewable via a standard web browser so that humans can explore, find, access, and understand. An advantage of this approach is that one can embed much information in the HTTP headers and in the HTML that make those data accessible to machines, including bots such as Google Search and data harvesters. So the endpoint of the data management is that the EMO-born data are published into web content and hosted for open consumption by anyone, any person, any machine. Okay, having spent quite some time and effort at developing the system, it is interesting to ask ourselves, what are the advantages and disadvantages of our approach to data management, and would we do it in the same way again? It's taken quite a lot of blood, sweat, and tears to develop the system, but nonetheless, I have to say the answer is yes. We would do it more or less the same way again. Given the needs and challenges we had, our approach works. First of all, we have zero cost for infrastructure. All the info we needed was that which already existed, either at our EMO-born partners or provided as a community resource. Next, the system is flexible and open-ended. If things change in the future, the system can adapt to those changes. The core interoperability layer that we add via the data model and the semantics and the row crates means that the inputs and outputs can change as data technology and specifications change, but the core functionalities that we have built do not need to. Another advantage is that we are not limited by any one system, one consumer, one need, one standard. We can choose what data or metadata we want to collect, which descriptions we want to include, regardless of whether that fits into a standard used by some particular resource. But we can also produce outputs that conform to standards used by others. The system we have built is the best way we can think of to make data findable by machines. And machine findability, accessibility, and interoperability are becoming more and more important with time. Next, the production of fair data is built in. Creating metadata and data that are findable, accessible, interoperable, and reusable is part and parcel of the system we have built because we make heavy use of semantics, machine-readable specifications, and web standard formats and schemas. However, on the minus side, we must note that there is a high cost on development time, on human resources. Developing the system required the order of a year of development time spread over the past few years. The actual development time is probably only about half of this. Analyzing what was to be developed, user testing, and going back and doing it again, that took the other half. But on a positive note, All the tools we have built in GitHub to do the data management are open access, and we are working hard on documentation. The system can be expanded or contracted depending on the needs of any new users who wish to adopt anything from this approach. We are very open to sharing our work. And that is the end of my presentation. I would like to thank the team, the Open Science team at VLIS, and I would like to thank the EMBRC people for helping me write this presentation and for developing the system. Thank you.